Now let's discuss about the chapter microbes and human welfare. So some of the important parts of this chapter is microbes are also beneficial to the humans and also not beneficial. But in this chapter we will be discussing about the beneficial perspective of the microbes. So we prepare Roquefort cheese through the fungi, then Swiss cheese through the Propionum bacterium Shermani, it's a yeast, and then Saccharomyces cerevisiae, it's for beverage industry also, and for bakers also called baker's yeast. Then Aspergillus niger from butyric acid from the Clostridium butyricum, Aspergillus niger acetic acid. Then from Aspergillus niger we get citric acid. From Acidobacter aceti we get acetic acid. Then in nitrogen fixing through symbiosis, example is Rhizobium, free living bacteria who do nitrogen fixing, Azotobacter, and Azospirillum. Then mycorrhiza is the association of fungi with the roots of higher plants. This association is done by genus Glomus, and it helps in the absorption of the phosphorus. So another cyanobacteria are Anabina nostoc oscillatoria. In paddy field, cyanobacteria serve as important fertilizer. Then cyclosporin A as immunosuppressant and derived from the fungus Trichoderma polysporum. Statin as lowers the blood cholesterol level derived from the yeast Monascus purpureus. Then streptokinase from streptococcus, and it work as clot buster. Then there is a treatment, millions of gallons of water treated every day, every day in whole world, and till now humans are unable to discover any machines efficient than microbes. So there is primary treatment, then there is secondary treatment, then secondary treatment also called the biological treatment. Primary treatment, in this first filtration is there, then after filtration there is sedimentation of the grit of the sand of the small pebbles, then which get sedimented, it's called the primary sludge, and the effluent and the is called, which is above portion is called effluent. Then effluent is taken to the secondary treatment or the biological treatment. In this, the effluent is now air is supplied, constant agitation is there, mechanically it's agitated. Then in this, BOD increases. BOD is the amount of oxygen needed by the microbes when it completely oxidizes the organic matter present in 1 liter of water. So as the BOD of the water decreases significantly, it indicates that water is somewhat pure. Then in this, the phlox grows. Phlox means mesh of bacteria and fungi, they grows. Now these phlox are sent to the, phlox are called, these are sent to the settling tank. Now anaerobic bacteria, so these are called activated sludge. Some part of this is again sent to the secondary or biological treatment tank, and others sent to the settling tank. Their anaerobic conditions, their anaerobic microbes grow, and so they die. And after secondary treatment, these are sent, water can be released into the rivers. So these were some of the points. And in village areas you will see gobar gas plant, 10 to 15 meter deep, 10 to 15 meter deep, and these were collectively made by the help of IARA, Indian Agricultural Research Institute, and KVIC, Khadi and Village Industries Commission. So microbes in production of biogas. So they also produce biogas, and which type of gas they will produce, it also depend on the substance they are growing or oxidizing. In fermentation of dough of cheese, they mainly produce CO2, but if the material is cellulose, then CO2 and hydrogen is the major. And in treatment, one point is missing, that is, and in anaerobic digester, their gases CO2, hydro H2S, methane, CO2, hydrogen sulfide, which is also gobar gas. So these are produced. Then Ministry of Environment and Forest initiated Ganga and Yamuna Action Plan. So we have heard about the secondary treatment plants, primary treatment plants. So these are some of the points of the chapter microbes and human welfare. That's all. Thanks for watching. Now some of the points of hydrocarbon. So preparation of the alkane. We can prepare it from the unsaturated hydrocarbon through nickel, platinum and platinum react over normal room temperature, but nickel needs temperature much slightly greater than the normal room temperature, and pressure also should be high. Then after this, from the alkyl halide, for this we need zinc and HCl, concentrated dilute HCl is needed, and except fluoride, alkyl fluoride, using this can't be changed into the alkane. Then by Duma method, then by Kolbe electrolysis method is there. Then oxidation of hydrocarbon will give you CO2 plus water. Complete oxidation, incomplete oxidation will give carbon plus water, carbon solid. Then incomplete oxidation in the presence of catalyst, like if we are using copper, 523 Kelvin temperature, 100 atm pressure, then it will convert into methane, will convert into methanol. If we are using molybdenum oxide, then it will convert into formaldehyde, and if we are using manganese acetate, then it will convert into ethane, will convert into acetic acid. So isomerization by the anhydrous LCl3 and HCl, aromatization chromium oxide, molybdenum oxide, and vanadium pentaoxide. Then reaction with steam. If a hydrocarbon will react with steam in the presence of nickel, if we will heat, it will convert into carbon monoxide plus hydrogen. Then pyrolysis. If you will do pyrolysis of hexane at temperature 773 Kelvin, nickel, platinum, then we will get hexane, hydrogen, then butene plus ethane, propene, ethene, methane. Then similarly we can do it of dodecane, nickel, platinum 773 or 973 Kelvin temperature, and will get heptene plus pentene. So these were some of the reactions. That's all. Thanks for watching.